Hello everybody. Okay, so in this video I'm going to be talking to you about monoclonal antibodies. So you need to know what monoclonal antibodies are and how they can be used in medicine, both as targeting medication to specific cell types or in medical diagnosis. You actually don't need to know about the process of the production of monoclonal antibodies, but you did learn about this at GCSE. So we'll spend a few minutes just reminding ourselves, because in any A-level question, they might expect you to have a basic level of understanding about this because it was something that you covered at GCSE, but it's not in the A-level spec. The other aspect that you need to know about, firstly, is how antibodies are used in something called the ELISA test, and we'll talk through the process of the ELISA test. There are two types of ELISA tests, the direct and the indirect ELISA test, and any ethical issues associated with the use of monoclonal antibodies. So we're going to talk through each of these points in turn. But first of all, we just need to talk about what a monoclonal antibody is. You already know this term, polyclonal antibodies. So we know that a single plasma B cell is capable of producing a monoclonal antibody, i.e. it's capable of producing only one type of antibody with one specific shape. But when we have a normal immune response, we produce many different types of plasma B cells, each producing its own specific antibody shape. So we might want some specific antibodies. And one of the reasons that we might want this is because antibodies have specific shape and they will bind onto specific proteins. So say, for instance, we wanted to detect or measure the presence of a specific protein in your blood, for instance, or in your urine. You could create an antibody that would do that, and you could test for things. For instance, you could test for pregnancy. So pregnancy tests test for the presence of something called the HCG hormone, human chorionic gonadotrophin, and it does like that by producing antibodies against this hormone. That's what is attached to the little strip in a pregnancy test, and we'll talk that through in a bit. You could test for the presence of particular antigens, for instance, antigens from HIV. That's how HIV tests work, or drug tests. So say, for instance, you wanted to test for the presence of a particular steroid or anything, heroin, whatever it was. That drug has shape, so you can create antibodies against that shape. So monoclonal antibodies can be really important for testing for the presence of something. The other reason they can be important is because imagine that you wanted to give somebody a drug to treat cancer, but that drug would kill any cell that it entered. So you want it to be specific. You only want it to enter cancerous cells. So scientists are able to isolate or identify the antigens that are present on our cancerous cells that are not present on other cells in our body, and then produce antibodies that are complementary to those cancerous cell antigens, and then attach that antibody to the drug. And then when you're injected to that drug, the drug will then only attach to the cell surface membrane of cancerous cells in your body. This direction of the drug means that firstly you can be injected with much lower concentrations of the drug, because it's not going to go through all the cells in your body. It's only going to go to this one specific tissue or organ where the cancerous cells are. And in turn, it means you're going to have far fewer side effects from this drug. So you're hopefully increasing the efficacy of the drug, so it's going to work better, and lower the dosage and lower the number of side effects. So it's a really beneficial treatment for something like cancer. So how do we actually get hold of monoclonal antibodies? Well, usually what we do is we use an animal like a mouse, for instance, and we would inject that mouse with the antigen that we were interested in. So if we were producing a pregnancy test, we would inject that mouse with the HCG hormone. This is foreign to the mouse's immune system, so the mouse will have a normal immune response. It will you have its phagocytes engulf and digest this hormone, and it will present the hormone on its surface of its cells, and then plasma B cells will be produced, and they will produce the correct shaped antibodies. Now, in the mouse's spleen, there are going to be really high numbers of these B plasma cells, so we sadly have to kill the mouse. This is a potential ethical issue that you might be asked to discuss in an exam question. You have to kill the mouse, and you extract its spleen. And then when you extract its spleen, you can then extract the B cells that are producing these antibodies. Now, these B cells are all well and good, and they're producing the antibodies that you want, but we know that one of the problems with differentiated cells in mammals is that they don't reproduce very much. They have something called the Hayflick limit. There's a certain number of times they can undergo mitosis, and then they stop, then they die, then they're broken down. So we want to get rid of that. So what do? We fuse these B cells with myeloma cells, with cancerous cells, because cancerous cells will just keep dividing rapidly and uncontrollably. So what we now produce is a cell that has the properties of the B cell, so it's able to produce the correct shaped antibody, and the properties of a cancerous cell, so it's able to rapidly divide. This cell is called a hybridoma. We then grow these hybridomas in a petri dish for a while, so we get loads and loads and loads of them, and they're all secreting out our antibodies, and we test them that they're producing the correct antibody that we want. And so we culture just that one specific type of hybridoma B cell, and it's pumping out all these antibodies, and then we can just harvest and purify these antibodies and use them for whatever we want to use them. And that process is the one that you learnt about at GCSE, so none of that should be new to you. So let's then talk about how these antibodies can be used in the ELISA test. The ELISA test stands for the Enzyme-Linked Immunosorbent Assay, ELISA. There are two types of ELISA, the direct and the indirect. We're going to talk about them separately. Let's talk about the direct ELISA first. In the direct ELISA, you use a known antibody to test for the presence of an antigen. So say, for instance, you suspect that somebody's bloodstream contains a particular antigen. For our example, it could be anything. Let's just say cholera. So let's say we think that they've got cholera, and we're testing for the cholera antigens. So we would have produced, using the method we talked about earlier, a known antibody that is complementary to the cholera antigens. So what would we do? We would adsorb it. We would stick it onto a surface. And then we would rinse that surface to remove any excess antibody that hasn't stuck to the surface. And then we would take our blood sample and we would add that to our surface. And what we would hope would happen is that if there were any antigens present from the cholera, they would attach to our antibody. We then rinse the whole thing again to remove any excess antigen that hasn't bound onto anything. The next thing that we do is we add an enzyme-linked antibody that is complementary to a different part of the antigen. I bet you can guess the next bit. We rinse the sample again to remove any excess enzyme-linked antibody. So we're rinsing the sample between each stage to remove excess. And even though that might seem a bit unimportant, it is actually important, because without that stage, it won't work. So the next thing we're going to add is a colorless substrate for the enzyme, and this substrate is going to be complementary to our enzyme. So actually, perhaps what I should have done when drawing this enzyme is given it an active site. So if the antigen was present in our bloodstream, when the sample was put on the antibodies, those antigens would have bound on to the binding sites on our antibodies, and then the enzyme-linked antibody would have then bound on to the antigen, and then when the colourless substrate came along, it would have bound on to the active site of the enzyme. And how do we know all this has happened? Well, once the colourless substrate binds on, the enzyme causes the substrate to change colour. And so we see it is visible. So the development of colour indicates the presence of the antigen. So this method can literally be used for testing for any antigen. And in an exam question, it doesn't matter what antigen they're asking you for, you just have to apply this method to it. Any question that you get asked could ask you to describe the process, but it's actually more likely to ask you to describe why you do each stage, so it's important that you understand both. So the second one is the Indirect Elisa. An Indirect Elisa is looking for an antibody, not an antigen. So for instance, say you wanted to know if you had coronavirus antibodies, so if you've previously been infected with coronavirus. You would basically do a very similar thing that you've seen to the Direct Elisa, except instead of attaching an antibody to the surface, you would attach a particular antigen to the surface. So in this case, an antigen to the coronavirus antibody. So let's just talk through those steps. So a known antigen is adsorbed to a surface. And then the surface is rinsed to remove any excess antigen. And then a serum, so for instance your blood or your urine, which is suspected of containing a particular antibody, is then added. And surprise, surprise, the surface is rinsed to remove any excess antibody. And then an enzyme-linked antibody, which is capable of binding to the constant region of the antibody, is added. Surprise, the surface is rinsed to remove any excess enzyme-linked antibody, and then we're going to add the colourless substrate of the enzyme. And when the colourless substrate of the enzyme is added, the enzyme will catalyse a colour change. That colour change will be visible, so the development of the colour indicates the presence of the particular antibody that you are testing for. And that's it. I know that's a lot of steps, but it's actually really logical. It's really straightforward. It's really simple. So hopefully you understood it. Go through it. Make sure you can draw it out. Make sure you understand each stage and why each stage is occurring. So in this video, we have talked about how we produce monoclonal antibodies, what a monoclonal antibody is, and we've also talked about the two ways that we can use monoclonal antibodies. So how we can use them for treating cancer by directing our drugs towards specific cells to prevent them from targeting non-cancerous cells, and also how they can be used in medical diagnosis, so through either the direct or the indirect ELISA. One other thing that the spec asks you to understand is the ethical issues surrounding monoclonal antibodies. And remember, in order to produce monoclonal antibodies, you need animal testing, and not just to produce monoclonal antibodies, but then to test them. So we need animal testing to check that those monoclonal antibodies work. So the issues are mainly around animal testing, but even when you have animal testing, when you then move into human testing, that doesn't mean that humans are going to react in exactly the same way as animals do. So for instance, in 2006, there was a drug trial where they were using monoclonal antibodies to treat arthritis. This drug trial resulted in organ failure in the human trial stage, even though there had been no problems in the animal trial stage. So you have to be really, really careful during drug development. And even though that drug had gone through the initial test with animals, it still caused harm to the humans in the that human trial stage of the testing. And so these issues have to be considered. You have to weigh up the risks and the benefits of developing a particular treatment. Okay, so that's enough for now. well done for listening. You need to go through all the processes, check you understand it, and then we will have a go at some exam questions.